Today we'll begin our talk about muscle tissue. We'll go through basics of muscles in general. And so when we look at muscle, it has some defining characteristics that we can see within the muscles and within the cells themselves. They are excitable, and so they have excitability. It doesn't take a whole lot for them to become able to contract. And so it takes just a little bit of, in a sense, irritation for that to occur. And that irritation comes from the neurons telling them when to contract. And they can contract, so they are contractile. And so they're able to shorten themselves. They allow the individual cells themselves to shorten, and that is what then starts to generate force. Once we have contracted them, if one muscle is contracting, another one is generally going to be relaxing and being stretched out. And as we do stretch them, they have the ability to stretch and rebound. And so they're not going to become damaged by that stretch itself. So they are also extensible. So we can uh, stretch them out and they have the ability to continue to contract even though they are fully lengthened. And so that allows for the muscle to do its normal job in a relatively easy fashion. When we look at muscle tissue itself, and kind of some of the things that it has going on for it, muscle itself generally we think of as being kind of movement. And so if somebody was to talk about muscles in general, they're going to think about them in terms of movement. And that movement is going to be either the entire body or uh, individual portions of the body or within the body moving things around, things like that. And so we do have different kinds of muscle tissues. And so they do have that ability to create movement kind of no matter where we are. As far as the whole body is concerned, we do have the ability to maintain posture. And so your head doesn't just fall over because muscles are pulling and contracting and maintaining that postural position that is there. Things like our abdominal muscles allow for support of soft tissues, and so they're protective to the underlying tissues themselves. When muscles contract, we've all experienced the process of working out and increasing our body temperature because of it. And so as muscles contract, they give off heat as a byproduct, and that heat helps us to potentially maintain body temperature. The muscles themselves as well have the ability to regulate the transport of material to different areas of the body. And so we have our smooth muscle, we have our cardiac muscle, and they allow us to regulate transport. For the most part, we're going to look at skeletal muscle as being kind of our general example of things. And so each muscle cell itself has the ability to contract. Individual cells we call either muscle fibers or the cells themselves. And so we have myocytes, we have these muscle fibers that have the ability to contract. Within a given muscle, those muscle fibers are then bundled up into what we call fascicles. And so we're going to bundle up individual muscle fibers into fascicles and kind of package them up. And then the whole muscle is going to have multiple fascicles inside it. And so it's going to have multiple components that are found within each muscle itself that's there. At the cellular level, at the level of the myofibrils themselves, we have what we call myofilaments. And so we have essentially the proteins that make up the individual components of a muscle cell. And so here we can see on the picture the different components that go along with the muscle. And so we have our individual cells. And so our individual muscle fiber that is there. And inside that muscle fiber, we have the different components that are there for it. And so here we have the whole muscle fiber. We have one individual cell. We have our myofibrils that are inside it. And then we have take one of those cells and put it into a fascicle. And so now we're going to take this cell and this cell, this cell, this cell, this cell, this cell, and so on, and wrap them up as well into an entire fascicle. And then the muscle then contains multiple fascicles as well. Each different component of the muscle is then surrounded by a connective tissue. And so we have endomysium surrounding the individual muscle cells. And so we can even see that here as the white line that surrounds those cells there. And then from there we have the fascicle surrounded by paramecium. And so we see that component that's there as well. And then surrounding the entire muscle itself, we then have epimysium. Surrounding the muscle, all of that connective tissue that's there, if you think about like at the store, you have steaks and things like that that are cut against the grain of the muscle fibers themselves, you can see those individual fascicles as being kind of like the connective tissue that's inside the muscle that's there. So all of that kind of marbling that occurs within beef is what you see as the actual fascicles themselves of the muscles and the connective tissue that surrounds. So back when we talked about skin, we talked about superficial fascia as being part of that hypodermic layer that we have there. And then deep to that, we then have deep fascia. And so this deep fascia is what covers around all of the muscles themselves. And so it kind of helps to hold together oftentimes muscle groups that do the same thing. And so they have a similar or exact same function themselves. And so when we look at things like the quadriceps muscles in the thigh, those quadricep muscles themselves are going to be surrounded by deep fascia. They're going to kind of hold them all together, even though they are four separate muscles. They're still going to be bundled up all together within inside that deep fascia that's there. It bundles them all together, and then it also kind of helps to separate each one from each other as well, so that they can move and slide past one another without causing any kind of friction between them either. When muscles attach, we think about them attaching to bone, and for the most part, that's what they do attach to. And so they're going to utilize a tendon, typically to go from the muscle fibers down to the bone itself, and then it's going to attach into the bone, or it may attach into skin directly. We have the ability to do facial expressions simply because those muscles are attaching directly into the skin itself. That's there. The tendons are made up of that dense regular connective tissue, so they're all kind of like a cord-like or a rope type of an appearance to them, and that allows them to pull on just a specific part of the bone, and that's what's going to lead to some of the bony structures that we have on bones themselves. So some of those bone markings that we see on bones are simply because of a tendon is pulling on those areas that are there. From there, we then have the ability to kind of create little sheets of connective tissue called aponeuroses. And so this may connect things like part of the muscle that covers your forehead back to a muscle that is all the way at the very base of the skull there, all the way at the back, the occipitalis. And so we have frontalis and occipitalis. More commonly, they're referred to as our epicranius muscle with a frontal belly and an occipital belly that is there. And so kind of goes by different names, and you may see that at different times depending upon who you're reading. Since the muscle has this tendon structure at oftentimes either end of the muscle, this is going to mean that we're going to have a distinctive, in a sense, kind of almost like a beginning and an end to the muscle. And so we have what we call the origin of the muscle, and then we also have the insertion. The origin is essentially the side of the muscle that the muscle is going to contract back towards. And so we're essentially moving the insertion towards the origin. And so we're pulling that part back towards the origin of the muscle itself. The insertion side is generally going to be the part of the muscle as well as the bone that's usually going to move the most. That's there. And so within our extremities, we have essentially oftentimes the origins and the insertions are kind of in two different places. And so the origin is going to be more proximal and the insertion is going to be more distal. And so we can kind of devise which one is which based upon where those muscles are. Muscle needs blood supply, and so it needs to have a fair amount of blood supply to it in order to keep it working. And so we get blood vessels that come into them. They're going to branch and branch and branch and branch as they come into the muscle, as they move through all of the different layers and go from the outside of the muscle into in around the fascicles until we get down in and around the individual cells, even with some of our smaller capillaries that are there. Nerves come in a similar fashion as well. And so an individual nerve is going to innervate that muscle, uh, regardless of where it is. And that nerve comes in and then it splits and goes out. And individual neurons then go and communicate with individual muscle cells and cause them to then contract. So here we have a muscle and a couple of different neurons. And so we have a motor neuron here that's coming into the cell. And so it's coming into this fascicle here. And what we end up seeing is that that individual neuron is going to innervate this muscle cell, this muscle cell, and this muscle cell. Those other three muscle cells that are there, these three here, are going to be innervated by some other neuron. And so we don't have, in a sense, kind of a situation where all of the muscle fibers are contracting necessarily at all exactly the same time, because this nerve that comes in splits and goes off to this one, comes off and goes to this one, and goes off to that one. And we have the ability to rotate, in a sense, when contraction occurs on either one. The motor neurons that do come in and all of the cells that it communicates with is what we call a motor unit. And so that individual neuron here, and then these three cells all together are our motor unit that we have there. And where it makes that connection there at that neuromuscular junction, all three of those cells are going to receive the information at essentially the exact same time. So all three of those cells will always fire off at the exact precise time when this neuron says it's okay to do so. Because skeletal muscle is voluntary, and so it has to be told when to contract. And more so, it's told when to contract when we consciously think about the process. So that motor neuron, and again, all of those individual muscle cells that it connects to, is the motor unit. And so this is kind of the basic contractile unit of muscle itself. And so one motor neuron, however many muscle cells that it actually communicates with, is that motor neuron. And oftentimes these are over 100 different cells that it's actually connecting to. The majority of muscles are not, in a sense, kind of all or one, in a sense. And so they're not just a single type of muscle where the whole muscle contracts all at the same time. It's not like a piston or something like that, or a robot, in a sense, where we're going to get all of the movement. And part of that comes from the fact that we have these motor units, is that we can contract only part of the muscle at any given period of time. And then the rest of the muscle can be relaxing. Then we contract another part of the muscle, and then the rest of the muscle is contracting. That's there. For the individual cell or the muscle fiber, that is definitely an all or none principle. And so for that, we have essentially contraction either happens or it doesn't. And with each individual cell, every time that it becomes stimulated by that neuron, it's either all contraction or no contraction. And so there's, in a sense, kind of nothing in between per se for that individual muscle cell itself that's there. In that neuromuscular junction, the communication between the neuron and the muscle cell is done so through a neurotransmitter called acetylcholine. And so acetylcholine is that communication between the neuron and the muscle fiber itself. And so that neuron comes along, it has little vesicles that we see inside there at the neuromuscular junction. Those vesicles then release acetylcholine out into the space here, and they bind to receptors, called acetylcholine receptors, and those acetylcholine receptors then cause the channels to open, and sodium can rush into the inside of the cell. And so we end up having the ability to have a large amount of sodium travel into the cell in a relatively short amount of time. The individual cells are further kind of packaged up a little bit further into what we call a sarcolemma. And so inside an individual cell, we have this sarcolemma, this space that's there in between inside a muscle itself. It has a large number of individual sarcolemma themselves that are there. The inside of the cell, when we talk about muscle, a lot of the things that are kind of common to other cells we're going to find here as well. But the main thing is that we're going to change the prefix on there, or the root of the word, to sarco. And so we're going to have a sarcolemma. This is the outer covering of the cell itself. And then we have our sarcoplasm, which is the actual cytoplasm that's there. And so this is the same as cytoplasm itself. We have endoplasmic reticulum, and that endoplasmic reticulum is, we change the name to sarcoplasmic reticulum. And so this is smooth endoplasmic reticulum. And its job is to store calcium inside it. And so it's going to store large amounts of calcium for when we want muscle contraction to occur. And so when we need muscle contraction to occur, we're going to release calcium into the inside of the cell. That's there. The sarcolemma isn't necessarily completely solid, so to speak. It has what we call T-tubules. And so the sarcolemma is kind of going along, and then there's a big long tube that goes down into the inner parts of the cell that's there. And that inner part of the cell has, I'll just make it green, has fluid that's on the outside, and that fluid that's on the outside is the same as the extracellular fluid. And so it gets it down deep into the inside of the cell. And then we have another one, and it comes down, and we have another T-tubule there. And that allows for us to essentially extend the electrical potential down deep into the muscle cell itself. And so it gets very far deep in there to where it can then join, and where we have a sarcoplasmic reticulum here. So our SR is there. And as our voltage changes along the surface here, as we change the voltage there, it eventually stimulates that sarcoplasmic reticulum to open up and dump all of its calcium into the inside of the cell. And so that ends up coming into the inside of the cell there, where we then have myofibrils. And so we now have our thick and our thin filaments, our myosin and our actin, available in order for us to cause muscle contraction. And so that's all happening at that neuromuscular junction. The sarcomere here is the space within a muscle cell where we're going to have that functional contraction occurring. And so by definition, a sarcomere goes from one Z disc to the next Z disc. And so we have Z discs at either end of a sarcomere, and then a new sarcomere begins. And then it goes to the next set of Z discs. And then the next one goes to the next set of Z discs, and so on. So within a single muscle fiber, we have many sarcomeres as well. That's there. And essentially, the sarcomere is the smallest contractile unit that we have. And so when we look at the sarcomere, we have a Z disc here, and we have a Z disc here, and that's the border of our sarcomere there. And then the next Z disc is over here, and then the next one there. So from here to here is a sarcomere, from here to here is a sarcomere, from here to here is a sarcomere. And all of those individual sarcomeres all end up contracting all at the same time. And when contraction occurs, the Z discs are going to be pulled towards the center of the sarcomere. And so they're going to get pulled in towards the M line there into the center of the sarcomere itself. We have a couple of other structures that we can see in a sarcomere as well. And these are the A band, the I band, and the H zone. And so the A band goes all the way from the edge of the thick filaments to the edge of the thick filaments. In a resting muscle, it's going to contain both thick and thin filaments. So the darker bands are our thick filaments, the lighter bands are our thin filaments. And so with an A band, it always goes from the edge of the thick to the edge of the thick. The H zone is the area here where we only have thick filament inside there, where we're only going to potentially have that combination of thick filaments in the very center there. From there, we have the I band. And the I band is the area that really actually spans two sarcomeres. And so it goes on either side there. And so it goes into partially one and then into the other. And so the two there are, it's on either side of adjacent sarcomeres. The I band and the H zone are going to change in size. The A band will always stay the same size whether we're contracting or relaxing. Those bandings that we see there in the dark bands and the light bands is what we end up seeing under the microscope as the striations. And so this is what gives the muscle its striations when we look at it under a microscope. And so we have the dark bands. These dark bands are where we have our A bands. This is where all of the thick filaments are. The light bands are where we have thin filaments. And so because they're not as thick, they're not as kind of hardy, they don't show up as dark on the microscopic view that we have there. And then within the sarcomere, we then have our I bands, our H zone, and the M line. So once again, the A band goes from the edge of the thick to the edge of the thick. The H zone is where we have thick filaments only. The I band is where we have thin filaments only within that sarcomere. And so once again, on a scanning electron microscope, we can then see those bandings that occur. We say that the A band doesn't change in size because of the sliding filament theory. And so when a muscle contracts, this helps to essentially define what is going on inside the muscle in order to get that contraction to occur. And so essentially the thin filaments are going to slide past the thick filaments. They're not going to change size, they're not going to change shape at all. They're just simply going to slide back and forth. And so when we look at a microscopic view of contraction of the muscle, we're going to see those bands start to change. In order for contraction to occur, we need ATP. So we need lots of energy here in order to do so. So when we look at that microscopic view as we're contracting that muscle, we can see that the A band is staying the same size. And so the A band here is the same length. The I band became smaller. And the H zone had become smaller as well. At maximum contraction, the H zone is gone. We can still see the A band, and the I band is gone. What should be over here is now gone as well. And so as muscle contracts, we see those differences within the cell's sarcomeres, and that gives us that sliding filament theory, that everything is just sliding past one another and nothing is really changing shape or changing sizes as it does so. We all know that when we have muscles they have some tone in them. They don't just feel kind of like jello or anything when we touch a muscle that we have. It has some tone to it. It has some amount of contraction that is already inside the muscle. And so some of the motor units are always contracting. And so we have these few amount of motor units that are basically always keeping the slack out of the muscle. It's kind of like if you had a rope or something and you need to maintain that slack in the muscle to where it's not really contracting per se, but it's just kind of keeping the muscle at the ready. So they don't produce any kind of movements, they don't produce any kind of changes. They're just simply going to cause the muscle to maintain that tone that's there and making it ready for the next contraction. When contraction does occur, in general, when we think of contraction of muscle, we're going to think of shortening of the muscle. The muscle is contracting, it's moving a load, it's causing a part of the body to move, things like that. As we get that contraction to occur, this is essentially the type of contraction that we have here is one that we would normally think of in the muscle itself. And so we can have an isotonic type of a contraction. And so we generate force, we're moving the load itself, and the muscle then starts to contract or starts to shorten. And so it's contracting, it's shortening at the same time. And as it does so, we get a change in size of the muscle versus a isometric contraction where that contraction occurs and there's no change in length. And so this is in a sense kind of like trying to lift something that is just too heavy. And so the load itself can't be moved. You're still generating force when you try and pick up that big rock or something like that. You go and trying to lift it, or you try and lift your car or something like along those lines. And you may move kind of the shocks and the struts and everything and kind of get them to move some. But as soon as it reaches its limit and you actually have to, in a sense, lift the car, you're not able to because you can't generate enough of a load there. And so that's limited by the size of the muscle that we have. The more muscle that is there, the more proteins that are there, the more readily we are able to do an isotonic contraction. So isotonic contraction, we move the weight. Isometric contraction, we don't move the weight. It stays still. It doesn't have the ability to move at that point. And so it's kind of like in the scenario here where we have one set of weights we're able to lift, and the other one we're just not. And so we're not able to generate that amount of force that's there. But isometric contractions aren't used just when, aren't essentially there only when we try and lift something that is too great. Isometric contractions also are used for things like posture. And so we're not trying to get movement, we're just simply trying to maintain the position of that particular joint at any given time. And so we have a series of muscles that are going to contract and cause that muscle to shorten, and we're able to do the movement that we want to. When muscle doesn't work the way that it's supposed to, if enough time goes by, we may get atrophy of the muscle itself. And so muscular atrophy occurs for one of two reasons. Either we take away the innervation to the muscle, or we start a process where we're not using the muscle the way that we used to. So we have denervation atrophy, or we have disuse atrophy within it. Either way, the outcome is similar to one another. And so that the muscle becomes more flaccid, it becomes weaker, it's not able to function the way that it normally does. This is part of what happens when somebody has carpal tunnel syndrome. So when somebody has carpal tunnel syndrome, they lose the muscle mass. And so here we have untreated carpal tunnel syndrome, and that whole thenar eminence there of the thumb is lacking any kind of real musculature on the hand itself. And so you can compare the two sides. This side, that part of the thumb really looks nice and kind of beefy, and this side, not so much. And that's just simply from untreated carpal tunnel syndrome. Muscle hypertrophy comes when we, in a sense, kind of overuse the muscle. And so we bring that muscle to a point where it is fatigued. It's basically saying, we can't do this anymore. We need more power. We need more strength. And that's where then we can eventually drain it, if even if we needed to, for that muscle itself, uh, for what's there. And so if we use the muscle over and over and over and over again, if we're fatiguing the muscle, making the muscle get kind of worn out, that causes that muscle then to get a signal at a nuclear level from the nucleus that says, make more muscle. And so each muscle fiber is going to develop more myofibrils. The myofibrils themselves that are there are going to have more myofilaments. That's going to lead to more mitochondria being used or built inside the cell as well. Potentially larger glycogen reserves coming about from it. Everything that makes that muscle stronger and bigger. With muscles, we have kind of different kinds of muscles, and so we can classify them based upon a couple of different ways. One of which is just to simply look at the fiber types that we have there. And so we can look at kind of the orientation of the ability of that muscle to actually contract. Does it happen very quickly, or is it a relatively slow process in terms of that contraction that's there in a sense? And so fast twitch types of fibers, these are the ones that we find that are moving things like our eyeballs. And so this is what moves our eyeball from side to side, up and down. If you ever actually watch somebody kind of come into a room that they haven't been to before, or maybe they even have been there and they're looking around for somebody that they know, you can watch their eyes and they're kind of darting all over the place. The eyes use fast twitch fibers in order to get that movement to occur very quickly. From there we have kind of intermediate fibers. And so fast twitch fibers have lots of glycogen. They have the ability to contract, and they have the energy that's there for the use of it, but they don't have as many mitochondria. They're going to fatigue after a little bit. They're not meant to keep on working and keep on working and keep on working. Intermediate fibers are a bit more fatigue resistant. They have the ability to kind of slow things down, so to speak. They're kind of the middle of the road as far as the muscles are concerned. Our slow twitch fibers are designed for long distance, long haul. And so these are going to be things like our postural types of muscles, the ones that we need to work all day long for the body itself. We can also classify muscles based upon their shape in terms of what we can see them looking like. And so we can have circular types of muscles. We'll see a couple of these when we start to look at the muscles themselves. So these are what are referred to as orbicularis types of muscles that we see around the eye and around the mouth. We have convergent types of muscles. Convergent types of muscles come down and almost oftentimes look kind of like a triangle. They're going to come down to a point. Things like the pectoralis major muscle, things like the deltoid muscle, they're all coming down into a single point as they insert. Parallel muscles basically are kind of flattened out muscles. And so parallel muscles are going to be things like our rectus abdominis muscle or rectus femoris muscle in our thigh, part of the quadriceps. And both of them have muscle fibers that are basically running along the entire length of the muscle. And so they're good for kind of tightening the region. Next up, we have pennate types. And so we have unipennate, bipennate, and multipennate. And basically what this starts to describe is basically how many heads does the muscle have. And so unipennate muscles are things like our forearm muscles. And so these are going to be things like our extensor digitorum of the fingers. And so extensor digitorum in our forearm. We have individual muscle cells and individual muscles, and they all converge down into a single tendon that comes down. Bipennate muscles have two origins. And so these ones are things like the muscles themselves go in two directions in a sense, and so this is similar to the rectus femoris again within the thigh. And then we have multipennate, lots of branches for them. Again, they kind of converge down into a single tendon there in the arm when we're talking about the deltoid, and so that comes down and inserts into the humerus. Muscles can work in a couple of different ways as well, and so we can have agonists, can have antagonists, and we can have synergists. Agonists are basically the muscle that does the most of the work for the action that we're looking for. And so for things like flexion or extension and things like that, whichever muscles is the biggest, strongest one when it comes to that particular movement itself. Those agonists oftentimes have synergists. And so synergists are going to help in the process itself that's there. They may be there more for stabilizing the joint as the other agonist is contracting. Antagonists are opposing muscles. And so things like our biceps brachii versus our triceps brachii. They're on opposite sides and they do different motions. When we name muscles, and so when we go to look at the muscles themselves, we're going to see that they come in lots of different names, and those can be based upon where they are on the body, and so the region that they're in. They can be based upon the shape of the muscle, the size of the muscle that's there. We can talk about the bipennate and multipennate type of state of the muscle as far as those things go. Sometimes the muscle just tells us is named based upon, in a sense, kind of what it touches. Which bones does it come into? And so that allows us to give names to the bones as well. So things like sternocleidomastoid, it tells us everything that it touches along the way. Biceps brachii tells us about the number of heads that muscle has, things like that. And so many different ways where the name origin of each of the muscles kind of comes from. And so again, some examples, things like supraspinatus. It's going to be found in the supraspinous fossa. So back when we did the bones on the scapula, we had the supraspinous fossa, and that gets eventually filled in now by the supraspinatus muscle. Masseter, it describes its action. It does mastication. It's chewing muscle. Cardiac muscle and smooth muscle are different from that of skeletal muscle. Cardiac muscle is only found in the heart, only in the cardia itself. That's there. And so these fibers are, as we saw when we did the histology of them, they branch. They do have striations, but the muscle cells themselves are shorter and they branch a lot within them. They typically have a single nucleus, but they may have two at times. The cells themselves here now are no longer voluntary. These are involuntary muscles. So as such, they also not only are they involuntary to where we can't control when they contract or relax, but we also can't control, in a sense, truly when they contract at times because they're autorhythmic. They set their own rhythm. They set their own pace in terms of how often they do contract. In between the individual cells, we also then have these intercalated discs. And so we have these special connections that we talked about before in between the cells themselves. And that also allows for gap junctions. We can move calcium in between the two cells. And then lastly, smooth muscle. Smooth muscle is spindle shaped. It's wider in the middle than it is at the ends. But when it contracts helically in that it twists around on itself. Imagine kind of wringing out a towel. And as it wrings out, it's going to twist and turn in on itself. And it's going to also thicken in the middle of that towel as well, similar to that of the muscles that are here. These can be autorhythmic as well, depending upon where they are. But they are definitely involuntary muscles that we have. These have a relatively slow contraction. So even the slowest of our skeletal muscles doesn't beat the slowness of smooth muscle. That being said, part of the reason why is that they tend to lack sarcoplasmic reticulum. There may be none to very little sarcoplasmic reticulum, which means that the calcium all has to come from outside of the cell. And we'll continue on into muscles the next time.